This is going to be Mendel's laws part 2, so we'll learn a little bit more about Mendel's law and a little more in detail. First off, we're going to look at the difference between a gene and a trait. And oftentimes these words are kind of used interchangeably, but they are very different from each other kind of in a sense, and they do relate to each other. So a gene is a unit of heredity coding for a trait. That's that DNA piece that's going to code for a particular trait. And we'll see that that gene is going to be transferred from one generation to the next. A trait is a genetically determined characteristic of an organism. So that particular trait is going to be that something from that gene is going to determine that characteristic. It could be eye color, it could be a particular disease. That gene could code for a trait that we're going to see as the outward appearance or the phenotype. When I'm talking about this here, then, I use a lot of different words that we'll need to know in genetics. And so I want to make sure that we're all on the same playing field. You might already know them and be like, this is a review. That's wonderful and great. You can fast forward the video. If you don't know these though, this is the number one reason why people usually get questions wrong is because they don't really know these words. So the first thing is an allele. An allele is a specific variation of a gene. Organisms are going to inherit alleles from both parents. And alleles are going to be represented with letters when we're making a Punnett square. So if it's a dominant allele, we're going to see a big letter A. If it's a recessive allele, we'll see a lowercase a. And so we'll see that a dominant allele always shows in the phenotype if it's inherited. So if the individual has a uppercase letter, they're going to then show that particular phenotype. Recessive is only going to show that phenotype when it doesn't have the dominant allele. And so that's when we only see a lowercase letter. So even if we see a lowercase letter and an uppercase letter, that uppercase letter, the dominant allele, will mask the lowercase letter, which is the recessive allele. Genotype is going to be the combination of inherited alleles. So we often say that that individual is big A, or little a. And what we're doing is representing if they're homozygous or heterozygous. If they're homozygous, their genotype has two of the same letters. So remember, homo means same, hetero means different. And so with heterozygous, they're going to be different in letters. You're going to have one uppercase and one lowercase allele. But again, that uppercase allele is going to mask the lowercase allele, and you're going to see a phenotype, which is the physical appearance of that particular genotype. And so, for example, the eye color could be brown. They could have really long hair. It could be even a particular disease as well. So those are all different phenotypes that it may express. We're going to now talk about this concept of the law of segregation. And what the law of segregation says is that chromosomes carry alleles. And we know that. We know that there's DNA inside here, and there's a particular trait or a unit of DNA that's going to correspond to a particular allele. And one is inherited from one parent, and the other is inherited from the other parent. And these chromosomes are going to be a homologous pair of chromosomes. And we know after meiosis that those are going to split into individual gametes from the mom and in individual gametes from the dad as well. And so homologous chromosomes carry the same allele. So we know that there's an allele here in black, and this allele here in black are the same exact trait that we're going to see on both of those. And so they're carrying the same allele. When the chromosomes are separated, though, into the daughter cells from meiosis, we're going to see that those alleles for the traits are going to be separated from each other. And the separation of alleles is what gives us that genetic variation. So we call this the law of segregation. It's the idea that these alleles are going to break into different spots so I should explain it this way. So in meiosis, we know that the chromosomes are going to split apart, and then in the gametes, we're going to see a different allele in each of those. We also will see here with the law of segregation. So here, let me go back one time. So here's law of segregation here. Then we're going to go here to the law of segregation again, but we're going to look at this with Mendel's experimental design. And so he often looked at P, which is the parent generation, F1, which is the generation from the parents mating, and then an F2 generation after that as well. And so what he noticed is that there was a reappearance of the recessive phenotype in the F2 generation, and that's evidence for the segregation of the alleles. So you can see here that there's a parent with big R, and little r, and we see no white flowers because all of these, we see that the big R is going to mask the little r in the phenotype, so they're all purple. But if we mate this generation, so this F1 and this F1, to get our F2 generation down here, you'll see that if both of these are heterozygous, then we will see that there are 3 purple and 1 white. And so this shows us that there has to be the law of segregation then occurring in this case. I'm going to go into independent assortment now. So we just did law of segregation, and now we're going to do the law of independent assortment. The law of independent assortment is that 2 or more genes are going to assort independently of each other. So one trait is not automatically inherited with another trait. For example, you don't automatically get being tall and brown eyes. Those don't come together. We are going to see that you could be tall or you could be short, or let's think of corn. You could have a purple kernel and a yellow kernel, but you don't have to have both of them. So we're going to see then that that trait is going to be automatically inherited with another trait. That's not the case in most cases. So that's the idea of law of independent assortment. And alleles for separate traits can be packaged into possible combinations in gametes. So for example, this trait here is independent of this trait here. And we represented it with a letter A and a letter B in this case. So when these chromosomes are going to go through the law of segregation, we know that you're going to end up with daughter cells, and we know that this big A could go this direction, this big B could go this direction, this big A could go here, this little b could go here, and it's this idea that you can inherit a big A without inheriting a big B, because you can see here you can have a big A with a little b. And so we see that they're going to assort independently of one another. It's not like if you are A, big A, you are also going to be big B. And if we say big A is brown eyes, I'm going to say, and big B is curly hair, you don't always have to have big eyes and brown hair. This is where, again, we see a lot more genetic variation than we would if this didn't happen. So we call this the law of independent assortment. So understanding Mendel's law of segregation allows us to use mathematical calculations. And this is for some people their favorite, or you really don't enjoy this. And this is where we're taking it from being more of a basic biology of understanding a Punnett square and now applying these math calculations to really figure out what is your chance of inheriting this particular disorder. This is something that genetic counselors often do. And it is a class that I'm hoping to offer next year is a genetics class. So if you are really interested in this, I would love for you to take this even though you're an AP Bio, unless you're a senior and you're off to leaving us at the end of the year. This would be something cool to learn a little bit more about. So look into that. It's going to be a genetics class that's offered next year. And so this is where we have our probability equation. The nice thing is they put the probability equation on the AP exam so you have it. The big difference, if it says if A and B are mutually exclusive, then you're going to use this equation. If A and B are independent, then you're going to use this equation. And we'll see that in just a second. And I'll also be doing practice with this in class with a worksheet and an activity. So just keep that in mind that we'll get into this in more detail. But I do want to kind of show you what it's going to look like. On the exam, you'll see this is your equation, just like we saw before. And we're going to look at a problem and just kind of see how this works. So assuming that genes A and B are not linked, okay? So that means that they're independent of each other. So we're going to be down to this equation here right away off the bat. If the probability of allele A in the gamete is 1/2 and the probability of B is 1/2, then what's the probability that both A and B are in the same gamete? We can just plug this in, 1/2 times 1/2, and in the end, we get 1/4 is going to be our answer there. The big thing is to look if they're linked. If they are linked, we are going to say that they are exclusive, mutually exclusive. They're in a relationship together. If they are not linked, we're going to say they're independent of one another. So a monohybrid cross, which is just the basic Punnett square that you've learned before, is an examination of how one trait is inherited. A dihybrid cross is an examination of how two traits are inherited. So that goes back to when we looked at the law of independent assortment. Sorry, law of independent assortment. You are going to see here that we have two traits. When we are doing a dihybrid cross, we're looking at two traits. A monohybrid cross is only one trait. Punnett squares are typically used to illustrate the probable outcomes of a cross. And so we can find our probability that way as well. Sometimes, though, on the test, it is easier to just use the probabilities, using the probability statements like this, than it is to go all the way through a Punnett square multiple times. So a monohybrid cross looks something like this, where you've got a big R, little r, little r. We carry down each of those, and we end up with two possible phenotypes, purple, dominant, and white, recessive. But in this case, from this, we end up with all that are going to be purple or dominant in this case. There are three possible genotypes. There's big R, which is homozygous dominant, little r, homozygous recessive, and heterozygous, which is big R, little r. Punnett squares show possible offsprings of a parental cross. So these are all the possibilities that we could get if we were to cross those two things. Oops, sorry, and I'm going to stop there. I will show you an example of a dihybrid cross as well. We will get into those. They're a little bit more tricky, but they're also kind of pretty easy once you get them started. If you are like, wow, I do not remember a lot of this, this is a lot of new words, you are going to want to slow down, make sure you have them in your notes, double check that you can at least do a basic Punnett square. I know it's been a little while, and just make sure that you're feeling confident on that, because we are going to pick up the pace this week. By the end of the week, we're going to get some more difficult, different types of Punnett squares, and we're going to learn a little bit more about dihybrid crosses, and we're going to apply that to another scenario. I will stop there for today.